The capacity of plants to survive in interplanetary situations with in-situ resources will determine how much of a contribution plants can make to improving human life support on other worlds. We use Apollo 11, 12, and 17 regolith samples to demonstrate that the earthly plant Arabidopsis thaliana germinates and flourishes in a variety of lunar regoliths. However, our findings demonstrate the difficulty of growing. Plants grown in lunar regolith developed slowly and several exhibited severe stress phenotypes. In addition, similar to how plants react to salt, metal, and reactive oxygen species, plants cultivated in lunar soils show differential expression of genes suggesting ionic stressors. Therefore, lunar regoliths are not completely harmful, and they can be used for plant cultivation in lunar homes. More research on plant regolith interactions is needed, and probably mitigation strategies before lunar regolith can be used effectively for life support in lunar outposts. Human exploration of the Moon's surface during NASA's Artemis Program 1 has piqued academic curiosity about the Moon's biology and its effects on Earth's ecosystems. Issues concerning the influence of the lunar environment on biology and biological systems have risen to the forefront of the lunar exploration agenda. The return to the Moon is envisioned as a devoted, longer-term commitment to lunar exploration. The inclusion of plants in lunar dwellings and exploring areas has been envisioned for some time. But until now, there had been no trials in which plants were truly grown in the authentic lunar regolith, and no one had even attempted to explore the interconnections between lunar materials and terrestrial biology in the era of contemporary molecular biology. Returning to the Moon for extended periods of time requires research into the effects of lunar regolith on terrestrial life and the viability of lunar regolith as an in-situ resource. Thus, we exploited plant growth and gene expression as a means of testing the basic influence of lunar regolith on terrestrial biology and providing an early assessment of regolith as a matrix for plant growth systems in lunar exploration habitats. Plant biology is an integral part of space research and development. The physiological adaptation of terrestrial biology to space exploration conditions can be better understood with the use of plants as model organisms, which give insights into gravity, radiation, and other space-related biological processes. Evidence of these developments is growing in the literature. Plants provide food and oxygen, recycle water, and remove carbon dioxide from human dwellings. These are all potentially crucial components to the long-term settlement of space and extraterrestrial environments. Since there is limited data on the effectiveness of employing in-situ materials, including planetary regolith, as a growth substrate, many designs of alien plant growing systems rely heavily on hydroponic systems. The ability to scale up the potential use of plants in lunar life support scenarios is facilitated by knowledge of plant development in the lunar regolith, which in turn illuminates fundamental biological interactions within the lunar environment. The Apollo moon landings marked a watershed moment in the history of space exploration. Not only because they marked the first successful visits to the Moon, but also because they marked the first time that terrestrial biology would come into contact with extraterrestrial material, and samples of that material would be purposefully returned to Earth. Protecting the astronauts on the Moon and the whole Earth's biosphere from the possible detrimental effects of returning lunar materials was an unusual worry after the first interaction between terrestrial life and lunar regolith. Besides poisonous mineral compositions and abrasive physical properties, novel, alien pathogenic microbes was the primary source of alarm. When the first Apollo crews returned to Earth, they were quarantined for 2 weeks together with the astronauts and medical professionals who had cared for them while they were away. This was done to safeguard the returning human explorers as well as the terrestrial ecosystem. In addition, at the Johnson Space Center Lunar Receiving Laboratory, materials were extensively separated in facilities using isolation protocols similar to those used in modern high-level biological containment, including the use of glove boxes. The primary objective was pathogen discovery. Pathogen discovery methods required direct, if brief, the interaction between lunar minerals and biology. Plants have never been cultivated in lunar regolith as the support matrix. However, lunar materials have been rubbed over leaf surfaces and dusted onto seedlings and growth medium. In the next study, we aim to fill in this informational vacuum. We investigated two questions. First, can plants grow well in the lunar regolith? And second, if so, what metabolic mechanisms were used to physically adjust to growth in this unique environment? These questions were approached separately and in tandem using differential transcriptomes. Arabidopsis was planted directly into Apollo 11 regolith samples, which were chosen for their diversity of composition. Samples from Apollo 11 and 12 are considered mature regoliths and submature regoliths, respectively, whereas the Apollo 17 sample is a combination of immature regoliths from different locations at the Apollo 17 site. All come from regions of the Moon where basaltic mineralogy predominates. For the Earth's reference standard, we brought in the Moon's mimic. The fact that the plants developed at all, despite obvious symptoms of stress, bodes well for future extended expeditions outside of Earth. Plants were said to have been grown for the first time on alien soil by scientists in May. 12 Arabidopsis thaliana seeds were planted in lunar regolith, which is the powdery substance that covers much of the Moon. This took place at the University of Florida's Space Plants Lab in Gainesville. The fact that they sprouted at all startled the scientists. Regolith on the Moon is quite angular and pointy. There was no evidence to suggest that plant life would welcome it. How quickly they expanded was the second shock. Plant development was stunted and under stress compared to that in soil on Earth. The results of the genetic studies shed light on the subject. The plants were able to physically adapt to their surroundings by activating several genes, despite their outward appearance. On the Apollo missions that NASA sent to the Moon between 1969 and 1972, astronauts gathered samples of lunar rock and soil. Since then, geologists have investigated samples to learn more about the Moon's history and makeup, but biological research has lagged behind. This is due to the fact that plants alter the soil in which they are cultivated, thereby rendering the samples useless. Regolith begins to undergo structural changes when air and moisture are introduced to it. The results of the experiment indicate that regolith might be of service in the cultivation of crops on the Moon. It demonstrates how vegetation influences the composition of soil on other worlds, which will be useful for future expeditions to the Moon, Mars, and other locations to look for traces of past plant life.